All right, so I'm going to just go over some of the things we've already talked about viruses just to begin with. And so viruses infect all living things, unlike the tree of life and all the kind of living things. There's no single origin. They're considered obligate parasites, and for the most part, not considered living things. And so, obligate means that they must be parasites. So it's the only way they can kind of exist and replicate. And so they rely on a host for replication to produce more copies of their own genes, genomes, and their proteins. Um, and the viruses themselves really have minimal kind of structure to them. Um, the biggest thing being there's no ribosomes, and ribosomes are the organelle that's important for producing um, chains of proteins from genetic information from nucleic acids. Um, okay, and so there's these kind of two things that all viruses have. One is those nucleic acids. So this is DNA or RNA that make up their genome. And then this is inside a protein coat. Um, and there's four different viruses shown here that have pretty different structures to them. Um, and this one shown down here is an animal virus that has a viral envelope. So some animal viruses will have this viral envelope around it that is essentially part of the cell membrane from the host. All right. Um, the protein coat is referred to as a capsid. So this protein kind of shell that will hold the viral genome. The capsids are made of capsomeres. So these are just little protein subunits that in many cases they'll just be one type of some subunit there's a bunch of copies of, and this is what makes the capsid. So down here we have tobacco mosaic virus. All the capsomeres are the same, identical to each other, and there's just thousands of copies that make the helical structure of the tobacco mosaic virus capsid. Um, we have another example over here with the adenovirus. This is icosahedral, which is essentially kind of like a 20-sided figure. Um, Again, this is mostly the capsomeres are all the same type, just identical copies making it up. There's a few things that are a little bit different. And here you can see these kind of spike proteins coming out. So those are slightly different than the repeated capsomeres. Here's a bacteriophage. T4 is the type of bacteriophage. It's a bit more complicated, complex, and so it's this icosahedral head, but then there's this kind of added tail portion to it, and it has a number, so 10 to 15 different capsomeres that make up this a bit more complex structure. All right, and then we can also talk a little more about the nucleic acid, so that genome, those kind of instructions for making more viruses. And so this can be DNA or RNA. And the DNA or RNA can be found as double strand or single stranded. And we'll talk a lot more about what this is and what this means in a little bit. And then the single stranded can be positive or negative, and that has to do with basically the coding versus the template strand. And so the viral genome, it's going to be both replicated and transcribed in the course of making more viruses. And depending on what the genome is, whether it's DNA or RNA, double or single stranded, makes a difference in kind of the process of how it is replicated and transcribed. And so one of the ways to classify viruses is based on the kind of types of genetic material. And here there's actually 7 in this classification scheme, but we just have 5 listed here. All right, and then these are the 4 main steps in all viral replication cycles, right? So the virus needs a host in order to replicate. The first step is the viral attachment, which is going to be the virus binding to the surface of the host cell. This is done in a very specific way. The second is viral entry, so the viral will enter the host cell. Once it's in, it's going to use the virus or the host cell machinery to make more copies. And then the pieces they self-assemble, and there's a few different ways that they can release from the host cell. All right, and then in the next videos, I will get into specifically these steps in bacteriophages.